So the primary objective at Sangamo right now is to apply the zinc finger protein technology. I think this is the only technology that allows you to work at the DNA level to knock out the major co-receptor for HIV virus, CCR5, and then we'll subsequently reinfuse these protected cells into the patients, which hopefully will provide them sufficient immunological benefit that will provide a functional cure in these subjects. At the time, we are trying to quantify the level of HIV reservoir in the subjects that we are treating, and we're struggling with the existing technology of a TaqMan PCR. The level of HIV DNA in a lot of HIV-infected subjects, surprisingly, maybe to a certain extent, is quite low. Even if we isolate the population of cells that the virus target, only 1 in 1,000 cells or so on average are where you'll find a copy of the HIV DNA. It's important for us to know what is changing in the patient in the reservoir, to measure that so we know what the drug is doing once we infuse the product into the patients. And so the QX100 technology is really a very useful tool for us in that we can use it to analyze the changes or fluctuations in the patient's HIV reservoir, allowing us to monitor what's going on once we have reinfused these cells into the patients. Or once we acquire the QX100, where we can subsequently repeat our measurements and see the same changes over time, we're a lot more confident in what the assay is telling us, is giving us something that's biologically real and relevant. Because if we're using a technology that generates data that you can really trust, the way the droplet help is that when you break up a sample into 10 to 20,000 droplet, essentially within each droplet you are doing a single PCR. So instead of doing one PCR, you're essentially doing 10 to 20,000 PCR, where only a fraction of all that 10 to 20,000 drop will contain your positive copy. So essentially what you did is you dilute out the background level of DNA, increasing your positive DNA to background DNA level to a point where you can more accurately determine the positive template that you're trying to amplify. It's fairly straightforward. It's not a very complicated procedure. There's a simple protocol that you follow. I think the first experiment we worked with our collaborator on this technology, we saw the result and we were amazed at how accurately and reproducibly they can measure low copy number events in a sample of genomic DNA. So the QX100 really is a godsend for us in a way that we can now more accurately measure the HIV DNA level or the HIV reservoir in these subjects.